Okay, well, let's begin. So I want to go over this review paper called Paving the Way for Adequate Myelination, the Contribution of Galectin-3, Transferrin, and Iron. So the reason why I want to go over this paper is because I've been a little bit on a kick on neurology, the galectins, galectin-3, and the galectin-3 binding protein explicitly, and iron and transferrin. And so this is one of the reviews that sort of brings a lot of that together for me. And I think that this is really important because in regards to COVID-19, COVID-19 seems to be able to screw up transferrin, iron, galectin level, or the galectin-3 binding protein levels, and a plethora of other things, but those are the ones that I think we should focus on, especially since COVID-19 and other coronaviruses seem to be able to cause diseases of the neurons, specifically or explicitly by attacking myelin. And myelin is the sheath, myelin is the protective coating around the axons of your neurons, which is what allows the electrical signals to basically pulse through them very efficiently. And so if you have these demyelinating events or the destruction of myelin, and this can be caused by a number of different things, well, if you have that happen, then you end up with neurodegenerative disorders such as multiple sclerosis. So, but your body is able to remyelinate or cause the production of new myelin, which can help prevent those progressive diseases from getting worse. So I think currently, in today's climate, this would be a very important subject to touch on because we might be dealing with an epidemic of neurodegenerative diseases as a result of this new coronavirus taking hold of this world, basically, and us just letting it happen. So without further ado, paving the way for adequate myelination, the contributions of galectin-3, transferrin, and iron. So let's begin. Considering the worldwide incidence of well-characterized demyelinating disorders, such as multiple sclerosis, and the increasing number of pathologies recently found to involve hypomyelinating factors. So hypo meaning too little, hypomyelinating factors are factors that prevent you from myelinating your neurons as readily. These factors, such as micronutrient deficiencies, elucidating the molecular basis of the central nervous system demyelination, and remyelination and hypomyelination becomes essential to the development of future neuroregenerative therapies. So being able to understand how these micronutrient deficiencies can screw you up, or being able to understand how we even get demyelinating events in the first place, coronaviruses being one of those, or remyelinate, and the understanding of remyelination, that will also be very important for understanding how we can develop these neuroregenerative therapies. So, in this context, this review discusses the novel findings of the contribution of galectin-3, also known as Gal3, and galectin-3, for those of you who are not aware or haven't watched any of my previous videos, galectin-3 is a lectin, and lectins are proteins that have the ability to bind and stick to some sugars that are on other things. So that's just what a lectin does. So they bind to sugars, or that's what they try to do. Okay, so galectin-3 specifically searches out the sugar galactose, explicitly the beta-galactose side. So when galactose has been covalently bonded to, say, a protein or a different type of biomolecule, the galectins will be able to, or the galectin-3 will be able to recognize beta-galactoside and then stick to it. Now, granted, there is a little bit of promiscuity with the lectins. Some of them can bind other carbohydrates a little bit to a degree, but they try to be specific whenever possible. So galectin-3 is named after its ability to bind to galactose. And for those of you who are not aware, galactose, so this would be the example of galactose, which is another one of the simple sugars, simple monosaccharides, and there is an explicit definition for what a sugar is. So it is a polyol that either has an aldehyde or a ketone group, so that the general formula is, I think it's a CH2O is the general formula for the sugars. So there are a lot of things that look like sugars but aren't. Now, what would a beta-galactoside be? Well, here we have galactose, and a lot of the sugars actually have the ability to form different types of ring structures, depending on, so here we have the linear form of galactose, and sometimes one of these alcohol groups are going to circle around and react with this aldehyde group that is on the galactose. So galactose has an aldehyde group, and other sugars could have, say, ketone groups. Regardless, this can react with itself to form these rings temporarily. And so in solution, these rings are forming and re-breaking all the time. Now, what Galectin-3 is searching for is it's searching for these beta D-galactopyranoses. So these beta D-galactopyranoses, this oxygen, this alcohol group right here, and I shouldn't call it an alcohol group, I should actually call it a hemiacetal group. Regardless, getting a little too far in the jargon, this is what the beta galactosides will look like. Is if this oxygen or if this bond is pointing upwards, and that would be in contrast to, say, an alpha galactoside where this oxygen is pointing downwards. So just so you know, when these sugars are being bound to proteins or other biomolecules, there is going to be a chirality or a direction that is associated with this particular hemiacetal group. Now, do not worry too much about it. Just know that this is a way to apply some specificity to the proteins. So that's what Galectin-3s try to bind to. They try to bind to those galactoses that are somewhere on, they can be on longer chains actually, so the galactose doesn't need to be directly on the protein. It could actually be part of a larger carbohydrate, but the principle is all the same. The Galectin-3s are going to search and destroy, or search and stick to these these galactosides. So anyways, we're getting a little off topic. So that's what Galectin-3 is. And then there is transferrin. So what is transferrin? Transferrin is one of the proteins that is able to allow the ferric ions or the iron 3+ ions to circulate throughout your body. And this is a way for your body to transport iron without causing damage. So granted, a lot of you might think that iron is in hemoglobin, right? And so the hemoglobin is circulating around the body. So why can't we use that for iron? Well, that's because the iron is in the hemoglobin, and so the iron needs to stay with the hemoglobin for the hemoglobin to work correctly. So the protein that is used to move iron throughout the body is transferrin. And there's another protein that is used to store iron, and that's called ferritin. And so these biomolecules are produced to sequester iron because iron is actually extremely reactive. And so since iron is reactive, it can catalyze a lot of unwanted chemical reactions while in the body. Some of those chemical reactions are reactions that happen with oxygen in your body, and it can form superoxide radicals, or it could form these very dangerous radical, or what is the word that's most commonly used? I think oxidants or radicals, yeah, free radicals, I think is the term that is used mostly in nutritive sciences. So these free radicals can float around and cause more damage within the body that is not just from the iron ions. So, and then iron can also cause just unwanted hydrolysis of your proteins. It can do a plethora of things. So that's why transferrin is, transferrin, that's why transferrin, this molecule, will bind to the iron to prevent it from happening. So with that, I think I'll just continue. So you have Galectin-3, transferrin, and iron, which all play a role in the process of myelination. And so what is myelination? So myelination is the, let's look up just myelin. So this is the neuron. And so this right here, this long part, is the axon. And the axon has these myelin sheaths that are surrounding the axon, and that protects it and helps the electrical impulses pass through more easily. And if you don't have those, you could have the situation like with people who are addicted to methamphetamines that actually can destroy a lot of the myelin sheath, and that prevents the electrical signals from flowing as quickly, and then you can have some disorders that can be the result of uh, the very unfortunate chemical dependency of methamphetamine. Anyways, basically it's a lipid or a very fatty substance that surrounds the nerve axons and insulates them. And let's see, I think that I don't have to go too far into it, but remyelination is if any of this gets damaged, you have cells in the body that are able, oh, okay, so myelin is formed from the central nervous system, formed in the central nervous system by the glial cells. So these are the non-nerve cells of the nervous system, if that makes sense. So these glial cells will sort of nurture the other cells, and they are able to form this myelin, or they create the wrapping for these nerve cells. So remyelination is caused by these glial cells. So the oligodendrocytes are the ones that explicitly do this. So let's go to the oligodendrocyte. And so here, this is an oligodendrocyte glial cell, and that will actually send out these little feelers and grab on to the axons of the cell, and then create these myelin sheaths that protect it from the outside environment, or actually both insulates it from the outside environment. So that's what these oligodendrites do. So in the process of myelination and remyelination, or I should say, in this context, the review discusses novel findings on the contributions of Galectin-3, Transferrin, and iron in the process of myelination and remyelination, and their potentially positive regulation of oligodendroglial precursor cells, also known as OPC. So remember, these oligodendrocytes, these are oligodendroglial would be precursor cells are the OPCs, and these would turn into these oligodendrocytes. So let's see if there's a Wikipedia page for this. So yeah, the oligodendrocyte progenitor cell would be the name of this, and I might actually use this for the thumbnail, right? So these oligodendrocyte progenitor cells will differentiate into these oligodendrites later to help cause or to allow them to be able to do these remyelination. So studies were conducted in the Cooper zone-induced demyelination and iron deficiency-induced hypomyelination, and the participation of glial and neural stem cells in the remyelination process was evaluated by means of both in vivo and in vitro assays on primary cells. So the Cooper zone-induced demyelination. So Cooper zone is a toxin, and this toxin is going to cause the myelin sheaths of the nerve cells to deteriorate. So let's see if there's a Wikipedia. So I think the only Wikipedia one is this demyelinating disease. So Cooper's zone. I really wish. Okay, just know that this is a chemical compound that causes demyelination events, and since it is a compound and it is reproducible to do this, we often use this in laboratory settings to have a reproducible form of demyelination. So let us see if Wikidata might be the best. Sorry. Looks like I'm just going to have to toss this because, well, what I can do is go to images. So just so you know, PubChem is pretty good, so I don't have to worry about that. So this is what Cooper zone looks like, and this will cause the damage to... Oh, and I think one of the reasons why it's called Cooper zone is that it is able to form these complexes with copper. So copper, right? So it seems to be able to form complexes with copper. So, which actually might be the way in which it causes neurodegenerative disorder, but I digress. So they give this cuprizone to the mice, and the mice end up getting demyelination events taking place. And they can actually, with iron deficiency, not having enough iron in the system, you can actually have hypomyelination because you do not have enough iron, and not having enough iron, or if you're anemic, you are unable to do this process effectively. So, and there is the participation of the glial and neural stem cells in the remyelination process. This process was evaluated by means of both in vivo in the living organism and in vitro in test tube assays on primary cell culture. So, with that, let's go. So the oligodendrocytes are glial cells in charge of myelin production in the central nervous system, and thus play a key role in the demyelinating disorders, among which multiple sclerosis appears to exhibit the highest incidence. Therefore, among the various trophic factors, so trophic factors, things that are trophic means that it's helping it become alive and grow. I think, let's just look up Google for what trophic means. So in relation to feeding and nutrition, in hormones, it's stimulating the activity of another endocrine gland, but let's see if there's a trophic factors definition. Generally be defined by any molecule that supports the survival of the cell. So anything that is supporting the survival of the cell would be considered a trophic factor. So therefore, among the various trophic factors which have been identified to support oligodendrocyte maturation and proliferation, for example, the fibroblast growth factor, the platelet-derived growth factor, the insulin-like growth factor, and thyroid hormones. This review will focus on the roles of galectin-3, transferrin, and iron in oligodendrocyte differentiation. So there's a lot of different growth factors that are associated with OLG maturation. However, in this particular review, they're going to focus their attention on the galectin-3, transferrin, and iron. So oligodendrocytes are known to produce most central nervous system transferrin. So transferrin, granted, transferrin is able to cross through the blood-brain barrier through the transferrin receptors. However, most of the central nervous system is getting its transferrin from these oligodendrocytes. So, and their maturation is directly related with the fact that the brain is the only organ in which transferrin mRNA expression increases after birth, compelling evidence of transferrin importance. So as you get older, your brain will produce more transferrin. It's upregulated in the mRNA level. So DNA gets transcribed into mRNA, and mRNA gets translated into the final proteins. So mRNA expression would be indicative of more protein being synthesized. So that happens in the brain. However, in the rest of your body, apparently you are producing less transferrin as you age, which I find very interesting. So perhaps that could be one of the roles in aging, is that you're producing less transferrin, which may or may not cause more labile iron to cause damage to your body. So that might be something to consider. In this context, numerous studies published by our group since 1994 have attempted to determine whether transferrin has an iron-independent trophic effect on myelin production. So here we know that if you don't have iron, you can have this hypomyelination, or you do not have enough myelination happening on your nerve cells. However, iron will upregulate transferrin, right? So iron upregulates transferrin, so they are asking, is it the iron that is necessary to cause this remyelination, or is it the transferrin that causes it, or is it both, or I should say, do you need iron and transferrin, or do you need just iron, or do you need just transferrin to have this effect? Or is it additive? Do they both affect it, or do neither of them affect it, right? So, well, it can't be neither because iron is doing something. Regardless, if you're able to get the same effect without iron, that might be an interesting finding. So, a critical question in assessing transferrin trophic action is whether apotransferrin, or ATF. This is transferrin that has no iron in it. So if you it's called ATF, but basically transferrin usually will have two iron ions that are associated with it, which allow it to get into the cell. Because when I believe as the transferrin binds to the transferrin receptor, it usually needs to be saturated with iron before it can be endocytosed effectively. Granted, do not quote me on that. So they are assessing whether if you just inject apotransferrin into a creature, is that apotransferrin going to have the same effect of, say, injecting transferrin with iron into the organism? So there's a lot of different variables that we have to correct for in biology in order to get a good understanding of the cellular signaling pathways. And they are assessing TF trophic actions is whether apotransferrin acquires iron once it is injected in vivo or from the cell culture media. So the problem with this experiment is if you inject apotransferrin, you can see these trophic effects, but is that because apotransferrin will bind to iron immediately and turn into trophic factor, turn into transferrin? Perhaps that's something that they're going to discuss in the review. So, and it suggests that it would rapidly bind to injected ATF even in small. Okay, iron ubiquity and its high affinity for apotransferrin suggests that it would rapidly bind to injected apotransferrin even in small concentrations. Although evidence seems to establish that it is apotransferrin and not iron binding to transferrin that has pro-differentiating effects. So they are suggesting here, and I believe their citation is of their own lab, 11 yeah, maybe. Yeah, E.F. Soto. Yeah, so they're quoting their same lab. So they are convinced, at least with their studies, that it is not the iron that is bound to apotransferrin that's causing the differentiation effects. It is just the transferrin molecule, or the apotransferrin, that is able to do this in of itself. So on the basis of the key role of iron in oligodendrocyte maturation and myelin production, and considering that hypomyelination as a consequence of iron deficits are associated neurological sequelae, or these are complications that happen after the fact, or sequels, they're sequel complications, they persist long after these deficits have been corrected. So people who have anemia, who get these demyelinating or hypomyelinating events from having anemia for extended periods of time, even after you add iron to their diets, they seem to still have these sequelae that are happening after the fact. So studies in our lab targeted the possible ameliorating effects of an intracranial injection of ApoTF in iron deficiency conditions, with results rendering a partial correction of myelin deficit. So that's very good to know, and that might perhaps be something that's important for us to, an important therapeutic that could be in the future. So subsequent assays in cultures of oligodendrocytes isolated from control and these iron-deficient animals revealed a smaller number of differentiated cells in the iron deficiency conditions, followed by a partial recovery upon apotransferrin treatment. Interesting. So finally, it was in recent years that our group has started working on molecules apparently working as external signals in oligodendrocyte differentiation, for instance, the galectins, which are well known within the immune system, but not as widely characterized in connection with the central nervous system. We have identified an essential role for the galectin-glycan interactions in regulating oligodendrocyte differentiation, leading to the control of myelin integrity and function. And in particular, we have assessed Galectin-3's potential role in modulating neuroimmune processes. So the Cooperzone and demyelination. So the Cooperzone model, or CPZ model, has been described as a useful tool to study demyelination and remyelination phenomena, with advantages such as easy reproduction, so reproducibility, that's something that we like to see in our tests, and low mortality rates. So don't kill your creatures. For one, they're expensive. Two, PETA will come after you. And three, you want to keep your death. It would be something that adds a lot of noise to your data, right? So you don't want to add more, what would the word be? You don't want to add biases, and having a large portion of your rats die would not be conducive to a reproducible and unbiased sample. So in the mouse models, a cuprizone diet has been shown to produce demyelination and oligodendrocyte damage in the central nervous system, particularly in the corpus callosum, or the CC. And let's, I don't know exactly where this is at, but it's a wide, thick nerve tract consisting of a flat bundle of commissural fibers beneath the cerebral cortex. So let's see if it has. You know what? I would not be able to parse this in a few minutes. So let us just say that it is a part of the. Oh, yeah. Okay. So here we go. So this is the corpus callosum, which is underneath the cerebrum, but it looks like it's above the cerebellum and above the, like, the hypothalamus and all those pineal gland, midbrain stuff. Regardless, that's the corpus callosum, and it's doing this it's causing damage to those cells without compromising other cell types, while its termination brings about the spontaneous and almost complete remyelination in a matter of weeks. So this is great to hear, that if you have some form of reversible toxin, then you can not only study the destruction of certain cell types, but you can also study the restructuring or the healing of those cell types to gain a bigger understanding, or a better understanding, on how these cells are talking with each other. So among although Kammer showed that cuprizone-induced cell damage in oligodendrocyte-enriched glial cell cultures and mixed glial cell cultures from neonatal or before birth rat, relating to just after, so newborn children. So shortly after birth rats, or the pups' brains, the demyelinating effects of cuprizone in vivo have been proven to be very effective in mice, but not in rats. Okay, so it's specifically targeting mice. So rats and guinea pigs exposed to cuprizone have been reported to show spongiform encephalopathy, but not demyelination. So yeah, just this would be sort of like CTE. So if you, that does not look good. Oh, so yeah, like this would be like a prion disease as well, but which would look similar to CTE. But just know that it is a disease that causes the brain to have a bunch of holes in it, like it is a sponge, right? So this is not demyelination, though. So it doesn't work in guinea pigs and rats. This doesn't work as a good model for demyelination events because it creates a completely different disease. However, it might actually have a role in creating this spongiform encephalopathy model as well. So perhaps we can use cuprizome in these animals to study, say, mad cow disease or CTE. Anyways, in turn, weaning Y-star rats, which is just a type of breed of rats, fed a diet containing a 0.5 to 2% cuprizone, they have exhibited intramyelinic edema. So edema means swelling, and intramyelinic, I don't know what that means explicitly. So I would just call it brain swelling, but let's look up the definition. So intramyelinic edema is caused by the lipophilic compounds that result in separation of the myelin laminae, or the sheets of myelin, at the intraperiod line. So in the central nervous system, this is due to the accumulation of fluid in the cytoplasm of oligodendrical cells. So if there is accumulation of fluid or edema in the cytoplasm of the oligodendrocyte cells, you can have this intramyelinic edema. So just know that this is swelling of that area, which can cause the myelin to sort of come off of the cells. So they have this and oligodendrocyte mitochondrial enlargement. So the mitochondria are getting larger. So among other abnormalities in different areas of the cerebellum. With these two exceptions, our group has pioneered the description of CPZ effects on myelin in rats. So similarity to what has been described in mice, CPZ demyelination in rats stops upon termination of CPZ administration and is followed by spontaneous remyelination. 2 weeks after toxin withdrawal, myelin yields increased substantially, and myelin protein, phospholipid, and galactolipid, so this would be a galactose that is bound to a lipid, contents recover. So, and let's see if there is, let's see if there is an explicit thing here that we should know. So galactolipids are glycolipids whose sugar group is galactose, and they differ from glycosphingolipids, and this is just another type of, it's just another type of thing, in that they do not have nitrogen in their composition. So here you can see there is a galactose, and remember what I was saying about this oxygen is pointing up, or it's pointing at us with this view, and so this would be a beta galactoside. And this beta-galactoside is bound to this glycerol backbone, and this glycerol backbone has these acyl chains, or these fatty, very hydrophobic chains that are coming off of it. So that's why it's called a lipid. And so this would probably, most likely, be an anchor into the cellular membrane. So they are main part of plant membrane lipids, so that's true, where they substitute phospholipids to conserve phosphate for other essential proteins, and that's really cool. I did not know that that was possible. So we know that the cells in your body, they are predominantly formed from phospholipids, where you have a phosphate group. So rather than having this galactose here, you actually have a phosphate group here. Now, granted, phosphate is a very important nutrient, and you might have heard for fertilizers, they have NPK numbers on them. So the N stands for the nitrogen content, the P stands for the phosphorus content, and the K stands for the potassium content, and those are very important for plants to get regularly. So phosphorus, since it is difficult for plants to get enough of it, they actually will replace some of their phosphorus in their cellular membranes with these galactoses. So that's, yeah, that's cool to know. So, and these chloroplast membranes contain a high quantity of monogalactosidal diacylglycerols, which monogalactose, so one galactosidal, diacyl, two acyl groups, or two fatty acids, and then glycerol. So glycerol is this middle piece. So there's that, and then there's also the diacylglycerol. And I don't know how they get another galactose on here, but they might be doing it by binding it to this original galactose. Regardless, I'm getting a little bit too into, if you haven't noticed, my master's is in carbohydrate chemistry, or carbohydrate biochemistry, so I'm a little bit more interested in carbohydrates than the average biochemist, but yeah, just so you know, that's what's happening. So they probably also assume a direct role in photosynthesis, but I think, oh, okay, so the galactosphingolipid, also known as galactocerebroside, and its sulfated derivative sulfatide is also in abundance present in myelin. Oh, okay, so this is what we're talking about. So this galactocerebroside, cerebro meaning of the brain, right? It is a ceramide, which I forget the definition, so a family of waxy lipid molecules. So it's a ceramide with a galactose residue at the 1-hydroxyl moiety. So yeah, just know that this is one of the things that is in the myelin sheaths, and these are a marker for oligodendrocytes in the brain, whether or not they form myelin. So these will exist in the oligodendrocytes, and they might form myelin later, but not necessarily. So yeah, and then remember how we were talking about, so the reason why this is called ceramide is that, for instance, with this MGDG, this was a glycerol that was here that was linking the two acyl groups together, or the two fatty groups together. Well, for the ceramides, that is not what they do. The ceramides are using a completely different backbone, and this would be the backbone that they use, which looks kind of like, well, it kind of looks like a glyceramine or glyceramide or glyceramine, so dihydroxyamine of some form. But yeah, this is just, they have only one acyl group, and then rather than having a acyl group over here, they actually have this completely different alkene group with a fatty chain at the end of it. So getting a little bit too far into the chemistry, I'm sorry. So this would be an example of what some of the sphingolipids are, but this would be one of the cerebrosides, where you have this, I believe, is galactose, but it doesn't need to be galactose. It could be a variety of other sugars. Now, let's continue. So 2 weeks after toxin withdrawal, myelin yields increase substantially, and myelin protein, phospholipid, and galactolipid content recovers. So remember, the galactolipids would be a biomarker of the glial cells. So to have more of it, or I should say, they are a biomarker of the oligodendrocyte cells. And so if you're having more galactolipids present, this is probably an indicator of having more oligodendrocytes, which would be good if you want to have remyelinating events. So, and although still remaining well below control values, so even though they are recovering, they are still well below what a healthy individual would have. So a suitable model to evaluate remyelination strategies in rodents without involving the adaptive immune system, the CPZ-induced demyelination of oligodendrocyte degeneration in white and gray matter is accompanied by reactive gliolysis, which is an increase in the number of resident microglia and, to a lesser extent, a rise in the number of peripheral macrophages. So what are the microglia? These are a glial cell, but they are also a immune cell. They are also an immune cell of some form. Now, granted, I don't know if they are derived from the bone marrow. So 1 second. Find bone. So yes, they are bone marrow derived and express high levels of the major histocompatibility class 2 proteins used for antigen presentation. So they are functioning similar to macrophages, which are a type of white blood cell, and they are from the bone marrow. So I believe they come from the hematopoietic stem cells. So bone. Okay, let's just read this. So another difference between the microglia and other cells that differentiate from the myeloid progenitor cells is the turnover rate. So yes, these are coming from the myeloid progenitor cells, which are bone marrow cells. Yeah. Oh, and so the dendritic cells and mast cells and osteoclasts. So yes, these are from the immune system. So you will have CPZ, or the cuprizone, will induce demyelination by the oligodendrocyte degeneration into the white and gray matter, but it's also accompanied by reactive gliolysis, and which is an increase in the number of resident microglia, and to a lesser extent, a rise in the number of peripheral macrophages. So you're getting more immune cells into the brain, which, for one, that's not normally a good thing, because immune cells can be destructive to the brain. For one, they could actually produce antibodies very easily against sensitive proteins and neural components of the nervous system. So that could theoretically be one of the reasons why you are forming multiple sclerosis, or these demyelinating events could actually be the result, or partially, of the immune cells creating antibodies against myelin. But I'm getting a little bit off topic, but just know that these people are trying to separate the effects of the immune system from the other effects. So here they're just trying to separate everything, which is sometimes difficult to do. So in order to characterize the mechanism of the cuprosome-induced demyelination, we used rat primary oligodendroglial cell cultures and evaluated the CPZ effects on cell viability. So this would be a way to avoid the immune system, is to just use a cell culture that does not have an immune system attached to it. And which was only significantly affected when either interferon gamma or tumor necrosis factor alpha were present. So, oh, interesting. So in order to characterize the mechanism of CPZ-induced demyelination, we use rat primary oligodendroglial cell cultures and evaluate the CPZ effect on cell viability, which was only significantly affected when either interferon gamma or tumor necrosis factor alpha were present. So this is interesting. So let's see what interferon gamma and tumor necrosis factor are, where they are coming from. So this is a cytokine, and it's critical for the innate and adaptive immune response against viral and some bacterial and protozoal infections. Interferon gamma is an important activator of macrophages and inducer of the class 2 major histocompatibility complex, which is one of the complexes or protein complex that will take antigens that these antigen-presenting cells will consume from the environment. They actually break up those antigens into smaller peptides, and then they will attach those to these major histocompatibility complexes and then present it on the surfaces of their cells for the other immune cells to see. So let's see where it is coming from. IFN gamma is produced predominantly by the natural killer and the natural killer T cells as part of the innate immune response. It is also by the CD4, TH1, and the CD8 cytotoxic lymphocyte effector T cells once antigen-specific immunity develops. So if you have some of these antigen-presenting cells that are, what would the word be? So if you have your T helper cells that are basically trained to go after a specific antigen, if they see this antigen that is on one of the antigen-presenting cells, then they will activate and multiply readily or rapidly. So IFN gamma is also produced by the non-cytotoxic innate lymphoid cells, which is a family of immune cells first discovered in the early 2010s. So that's interferon gamma. So we know that interferon gamma is coming from immune cells. I think that's what we're trying to understand here. So, and tumor necrosis factor alpha, where does that come from? So, and I've read these things like hundreds of times and I forget them every single time. So I have to since there's so much in biology, I always have to, refresh my memory. But this is a cell signaling protein, which is also a cytokine involved in systemic inflammation, and it is one of the cytokines that make up the acute phase reaction, which the acute phase reaction is a class of proteins whose plasma concentrations increase or decrease in response to inflammation. So it is produced chiefly by activated macrophages, although it can be produced by many other cell types such as the T helper cells, the natural killer cells, neutrophils, mast cells, eosinophils, and neurons. So neurons can produce this tumor necrosis factor alpha. So going back to this CPZ effect will not kill or not even have much of an effect on these oligodendrocyte cells unless you have interferon gamma or tumor necrosis factor alpha added to those oligodendrocyte cells. Okay? So this INF gamma can only come from the immune system, or it predominantly comes from the immune system, and tumor necrosis factor alpha is predominantly coming from the immune system or neurons, but we don't have any neurons in the cell culture. This is just oligodendroglial cells. So if you only have the oligodendroglial cells, you will not see this effect unless you add something that should be coming from a different cell. So In addition, in vivo experiments showed that the inhibition of microglial activation with minocycline prevented CPZ-induced demyelination. So, what is minocycline? Search Google. So, sold under the brand name Minosin, this is a tetracycline, tetra meaning 4, cycline meaning there are 4 rings, used to treat a number of bacterial infections such as pneumonia. So it is also used for treatment of acne and rheumatoid arthritis. The side effects, now let's look at the, see if it has a mechanism of action here. So it's just an anti-inflammatory effect, and they use it in multiple sclerosis and rheumatoid arthritis and Huntington's disease. So it's doing that. So it inhibits nitric oxide synthase indirectly. So it indirectly inhibits nitric oxide synthase. So let's see if there's anything here that we should study. Yeah, I don't think there's anything here that I really should go over. Just know that this is something that can suppress the immune system to a degree. It's anti-inflammatory, but it seems to also be good against diseases too. So I won't be able to tell you exactly what this does, unfortunately, without reading more. But so they add minocycline, and it prevents the CPZ-induced demyelination. So taken together, our results demonstrate an active role for microglial cells, which are immune cells, in the CPZ-induced oligodendroglial cell death and demyelination through the production and secretion of pro-inflammatory cytokines. So it appears as though these pro-inflammatory cytokines are absolutely necessary for these demyelinating events to happen, because if you are giving CPZ to these glial, oligodendroglial cell cultures, nothing happens to them, or nothing bad happens to them. But you need to do that in combination with these pro-inflammatory cytokines to cause this demyelinating event. Now, granted, what this is saying to me is that what's probably happening is your brain will have antigen-presenting cells, right? So these, and I've done another video on this, so if you want, you can go back and look at my major histocompatibility complex video that discusses what is going on with these antigen-presenting cells. But basically, things such as the dendrocytes, or the dendritic cells, I should say, of the immune system, or other antigen-presenting cells, they are consuming stuff in the environment. Now, when everything is happy and you are not inflamed and you do not have the signal that things are bad, these antigen-presenting cells will be happy. And so they present antigens, but the antigens they present will be associated with a healthy state in the body. So that tells the immune cells, do not attack these antigens because everything is good. However, if you go into a very pro-inflammatory state, like what is happening here with cuprizone, then what ends up happening is those same antigen-presenting cells will go from being benign into being turning into their mature form. And in their mature forms, they are presenting those same antigens, and then the other cells around them, other immune cells, will say, oh crap, these antigens are bad. I need to attack these antigens, or I need to develop antibodies against these antigens, or I just need to search and destroy, right? So when it comes to your brain probably has these antigen-presenting cells living normally happy lives in your brain. Probably these microglial cells are probably, let me see if these microglial cells are antigen-presenting cells. So they are a specialized population of macrophages that are found in the central nervous system, and they remove damaged neurons. So for one, they remove damaged neurons, right? So they are already going to be consuming a lot of the stuff that is within the brain, and infections are important in maintaining the health of the central nervous system. So let's see if it is antigen Ah, yes. So due to the lack of antibodies from the rest of the body, this is good. This is good for everyone to know. So due to the lack of antibodies from the rest of the body, so few antibodies are small enough to cross the blood-brain barrier. So that's one problem. The microglia must be able to recognize foreign bodies, swallow them, and act as antigen-presenting cells for activating T cells, right? So yeah, these microglia are the antigen-presenting cells which activate T cells. Good to know. Now, granted, I'll leave it at that. There might be more to this that we have to come to later. Just know that that's happening. So, taken together, our results demonstrate an active role for the microglial cells in the CBZ-induced oligodendroglial cell death and demyelination through the production and secretion of pro-inflammatory cytokines. So these pro-inflammatory cytokines are going to possibly activate these microglial cells or get them thinking that the antigens that are present in this environment are dangerous. And so that can be very negative. That can be very negative for your brain because a lot of the brain's antigens might be presented on these microglial cells in the major histocompatibility complexes. And if the brain components are being presented as antigens and you're in a pro-inflammatory state, that might inadvertently activate these T cells into attacking the brain tissue, right? So you want to avoid this if possible. So that's good to know. And then there might be the other situation where you actually do end up forming antibodies against these. However, that would require not the T cells, but different sets. I keep on forgetting antibody producing. Yeah, so they are B cells. The B cells are produced by the specialized white blood cells called B lymphocytes. So, and then when the antigen binds to the B cells, it stimulates the B cells to divide and mature. So a B cell might actually interact with an antigen-presenting cell as well, and that could cause them to be stimulated. So theoretically, it is possible that a B cell might actually make its way into into the brain, into the nervous system. Unfortunately, I don't think this happens frequently. I'm spitballing right now. I'm spitballing things that are possible. So if a B cell were to theoretically get into the central nervous system, somehow sneak past the blood-brain barrier during a hyperinflamed state, which is possible, then if it were to interact with, say, a mature microglial cell that is presenting antigens that are coming from the brain, then theoretically, if the wrong B cell were to get in, it might be able to start producing antibodies against brain tissue as well, which could theoretically cause a neurodegenerative disorder down the road. So let me see if there's anything that I should really focus on right now. Nah, I think for now I'm going to leave it at that, and then we'll get more into it later in a different video. So the demyelination, or the demyelinating diseases and oligodendrocyte damage, endogenous sources for remyelination. So demyelination is defined as the process governed by the loss of myelin sheets around the neuronal axons, and it is a consequence of oligodendroglial dysfunction or death, which, if sustained in time, inexorably leads to neuronal damage and neurodegeneration. So among human demyelinating pathologies of the central nervous system, multiple sclerosis has a mean age of onset in youth, adulthood, and multiple sclerosis has a mean age of onset in young adulthood and clinical neurodegenerative manifestations in motor, sensory, and cognitive disability. So multiple sclerosis is considered a chronic autoimmune disease and is characterized by the emergence of demyelinating foci in different brain areas, which is accompanied by a marked inflammatory response with microglial activation and astrogliosis. So what is astrogliosis? So the astrocytes are another type of glial cell, and astrogliosis is an abnormal increase in the number of astrocytes due to the destruction of nearby neurons from the central nervous system. So just so you know, that's what the astrocytes, or that's what astrogliosis is it's an excess of the astrocytes, which are another or a different type of glial cell. So with that, although the adaptive immune system plays a leading role in the acute pathogenesis of multiple sclerosis in most cases, the primary oligodendropathy prevails in some patients. An endogenous tissue response to central nervous system demyelination, remyelination, tends to restore oligodendroglial populations and myelin sheaths and is detected at an early stage of multiple sclerosis. So, in the early stages of multiple sclerosis, we can have this central nervous system demyelinating event, but immediately we have remyelination. We are trying to remyelinate, so we create more of these oligodendrocytes to help with the remyelination process. However, in the long run, the process fails. The process fails in the long run, and the pathological environment associated with the disease prevents successful long-term myelin recovery. So whatever we're doing, so in multiple sclerosis, we are trying our best to fight against this demyelination event. But it doesn't prevail. It doesn't do it fast enough. It doesn't do it well enough. And so even though you are ramping everything up, you still end up failing in the end. So to me, and that would be the adaptive immune response. That would be the response of, say, antibodies being generated against something within the brain, right? So this is why I'm a little bit worried about coronaviruses, right? Because for one, the coronaviruses can cause inflammation of the brain. They can actually travel through the neurons from one neuron to another through diffusion or by transfer through the axons, but they can transfer to deep parts within the brain. And then they can actually do biomimicry, or they can mimic some of the proteins of the brain, which is awful because if you form antibodies against the virus, you could inadvertently develop an antibody against some of your peptide hormones, or you can develop antibodies against the receptors of those hormones. So one thing that a lot of people aren't aware of is you have what is referred to as anti-idiopathic antibodies. So anti-idiopathic antibody. So let's see if I can get I might just have to go to another website. So this is what an anti-idiotypic antibody is. So you have one antibody that binds to an epitope on the antigen. And so this would be an idiotype that binds to this particular antigen. Now, unfortunately, you can have an anti-idiotype antibody. So these are an antibody that is actually able to bind to the original, or bind to the antibody. So you can actually form an antibody cluster this way, where they actually will form chains of these antibody and anti-idiotype antibody over and over again. Okay. Now, something that could theoretically happen that would be very unfortunate is, let's say you form an antibody against a peptide, right? Or a hormone, if you will. Let's say you form an antibody against a hormone. And I've done another video on this. So if you want to go into, I just do a search of my channel with the antibodies, and there will be a list of different videos, and there will be one of them that goes over this particular topic a little bit more. But basically, the first antibody could go against a peptide hormone. And then you can actually generate an antibody against the first antibody. Now the problem is that second antibody will possibly look very similar to the peptide hormone, which then means that whatever receptor that peptide hormone were to go into, the antibody might also be able to fit into that same receptor. And so you run the risk of actually generating antibodies, not just against your peptide hormones, but against the receptors that those peptides act upon. So just be aware that this is something that theoretically could happen in the brain. And I'll leave this up because I will put this into this description if you want to go to that webpage yourself. So, sorry, I have a lot of tabs right now. So much of the effort has been, oh, so talking about multiple sclerosis again. So what it's sounding like is that in multiple sclerosis, there seems to be an initial pro-inflammatory demyelinating event that takes place. And then as a result, your body tries to fix that demyelinating event by remyelinating your neurons. And however, the problem is that event somehow has caused continuous progressive degeneration. So even though your body has responded with a remyelinating event, that remyelinating event won't be able to run forever. It won't be able to counteract this progressive disease. And so eventually the process fails and you end up with multiple sclerosis. So, however, much effort has been made lately in order to understand the pathophysiological complexities that unfold during multiple sclerosis, and attention has been drawn towards developing effective strategies to stimulate myelin repair in the injured brain. In this context, although animal models do not completely recapitulate human multiple sclerosis features, they continue to stand as useful tools for studying remyelination processes in vivo. So even though these animal models cannot fully model human multiple sclerosis, we still use them to try to understand how the remyelination process, what proteins are associated with the remyelination process, what nutrients are necessary, what is the immune system responsible. All these things we can learn from these animal models, even though it's not a perfect fit. So in the Cooperzone-induced demyelination models in rodents, several hormones such as thyroid hormones, sex hormones, or differential growth factors have been shown to stimulate myelin recovery. Despite their effectiveness in promoting remyelination, the exact mechanisms through which these factors trigger oligodendroglial generation are still unresolved. So remyelination could proceed by two alternative mechanisms: a rapid response of local oligodendroglial precursor cells, also known as OPCs, which undergo differentiation and oligodendroglial maturation, and B, a process involving the neural stem cells. So you could have these precursor cells, which in of themselves, they're multipotent, or they have potency in the ability to differentiate more, or it could also involve these neural stem cell activation and neural progenitor cell proliferation from the subventricular zone niche. So what is the subventricular zone? I do not know. It's just a region situated on the outside wall of each lateral ventricle of the vertebrate brain, and it is present in both the embryonic and adult brain. So just know that it's a part of the brain. So, and then there could be migration from this subventricular zone niche. You can have the migration of these progenitor cells towards the damaged brain areas and terminal oligodendroglial differentiation. So it is possible that those progenitor cells end up migrating to the area of damage and then turning into the oligodendroglial cells, where they can then repair the neurons. So in vitro studies are useful to dissect these particular mechanisms and allow puzzling out cell interactions and evaluating the effects of individual factors on specific cell types. Oligodendroglial-enriched primary cultures have been extensively used as an in vitro model of oligodendroglial maturation. So more recently, NSC and NPC biology research has come as an exciting, and this is the neural stem cell and the neural progenitor cell biology research has become an exciting field of study within neuroscience, where multipotent neural precursors can be manipulated in vitro to obtain OPC-enriched primary cultures. So that is wonderful. Now, granted, there was recently a paper that came out of turning astrocytes into neuron cells based on different types of factors that they added to it. So, okay, so with all of that background, we can finally get into the reason why we started the video, which is the Galectin-3. So galectins, these are a family of beta-galactoside-binding lectins lacking specific individual receptors. So they don't have their own individual receptor. They can actually bind to a lot of different things that have these beta-galactosides on them. And I should also mention, theoretically, there are some peptides that can actually imitate or mimic carbohydrates, so it is possible that there are a few peptides, or at least amino acid sequences that might also be able to bind these galectins through the same mechanism. Anyways, they bind to the cell surface glycoconjugates containing suitable oligosaccharides to form multivalent complexes and induce intracellular signals regulating cell survival and differentiation. So these galectins, they bind to something on the cells, and then frequently those things are on the cell surface, and since they're binding to different receptors or different cellular components, there's usually some sort of downstream cellular signaling that is taking place. Anyways, and for one, the Galectin-3s, one such example would be they can actually kill T cells. They can find a way to convince a T cell to kill itself or undergo apoptosis, right? So, you know, going back, let's say that going back to, say, the microglial cells. The microglial cells, I should take that back, but the microglial cells will produce, might theoretically activate some T helper cells, which then the T helper cells then could theoretically damage the brain tissue. But I think Galectin-3 only works on the immature T cells or the thymocytes, so I don't think that Galectin-3 would theoretically. I'll have to look at that. I think in the process of maturation, the T cells become resistant to Galectin-3, but I did another video on it, so you can go back to that if you want to watch it. So, anyways, the galectins also form complexes that cross-link glycosylated ligands to form dynamic lattices. So these are called Gal-glycan lattices. So, and Gal-1 and 3 often have antagonistic roles in the immunological system. So while Galectin-3 plays pro-inflammatory roles, Galectin-1 exerts anti-inflammatory effects. And this is a general rule of thumb, but there's so many exceptions in biology, I don't know if I can actually take this phrase at face value. Anyways, Galectin-3, which is a chimeric protein structurally composed of unusual tandem repeats, so these are repeated sequences that come right after each other, of proline and glycine-rich short segments fused into a carbohydrate recognition domain, also known as a CRD. This possesses multifaceted roles in physiological processes, including the regulation of the innate and adaptive immune responses. Specifically in the central nervous system, our group was a precursor in establishing the relevance of the galectin-glycan lattices in oligodendrocytes, OLG, this is the oligoglial physiology, and identifying an essential role for the lectin-glycan interactions in regulating oligodendroglial differentiation, leading to the control of myelin integrity and function. So these lattices seem to be important in regulating this differentiation. And after this differentiation takes place, then they are able to create the myelin, right? So our first findings showed one second. Yeah, sorry about that. Should probably stay hydrated. So, oops. Sorry about this. I lost my screen. So our findings showed a high expression of both Galectin-1 and 3 in the astrocytes, which are another type of glial cell, and also in the MGs. And I don't remember what MG is, so MG neurocell. The myasthenia gravis, which is no, that's not what I want. Oh, the microglia. Yeah, so yeah, the MG is microglia. So the astrocytes and the microglia both have Galectin-1 and 3, and an upregulation of Galectin-3 in differentiating into the oligodendroglial cells. So as there is differentiation into the oligodendroglial cells, there is a big upregulation of Galectin-3. This upregulation was accompanied by an increment in the activity of metalloproteinases. So metalloproteinases are enzymes that can break down other biomolecules, and they use metals as the important cofactors. So let's search this up so that we have an understanding of that as well. They are protease enzymes, and I should have said that they explicitly will break up other proteins, but they're protease enzymes whose catalytic mechanisms involve a metal. So yeah, just know that this is important. So you have an increase in these metalloproteinases, which are in charge of regulating the biological activity of galectin-3 during oligodendroglial differentiation. Interesting. So, interestingly, recombinant galectin-3 treatment accelerated oligodendroglial differentiation in a dose and carbohydrate-dependent manner, which is in accordance with the N-glycosylation profile observed in immature versus differentiated oligodendroglial cells. Interesting. So, there seems to be both a carbohydrate requirement and a dosage of galectin-3 requirement, which allows or forces the differentiation of these oligodendroglial cells. So strikingly, OPC cultured, these are the progenitor cells, so the oligodendrocyte progenitor cells cultured in media conditioned by galectin-3 expressing microglia produced a higher number of differentiated oligodendroglial cells than the OPC incubated in the presence of galectin-3 deficient microglial cells. So these microglial cells, these are some of the immune cells of the nervous system. You can mutate them so that, well, you could either mutate them or you could force them to either express or not express this galectin-3. And if they are forced to express it, you grow them in culture with these the progenitor cells, it actually allows them to differentiate into these oligodendroglial cells more readily than without it, right? So these findings received strong support from our in vivo results. The morphometric analysis of the ultrastructural studies revealed a significantly lower number of myelinated axons and myelin turns, or also known as lamellae. So remember, this myelin sheath is wrapped around over and over around the axons. There was less of that in the galectin-3 negative mice. So mice that could not express this ended up having less myelinated axons and less myelin turns. So furthermore, the G ratio, which is the ratio between the axon's diameter and the axon's diameter wrapped with myelin, this was observed to be much higher in the negative control. So basically, the axon is thicker compared to the rest of the myelin sheath in the negative ones, because the negative ones can't produce as much myelin sheath. However, if the mice do produce the galectin-3, then they produce more myelin, so the ratio should be smaller. So accompanying these results, behavioral studies in the galectin-3 negative mice rendered lower anxiety levels, which resembled those observed during early CPZ or the cuprizone-induced demyelination. So anxiety of mice would be an indicator that the mice is very attentive or hyperattentive to its environment. So if a mouse has too low of anxiety, that's definitely not a good thing or a healthy thing. And they see this in the early stages of the demyelinated rats or the demyelinated mice. So this would be an indicator that being galectin-3 negative or not having galectin-3 would cause similar demyelinating or similar lack of myelination that you see in the demyelinated mice. So finally, neurospheres isolated from the LGAL3 negative mice showed a weaker commitment to an oligodendroglial fate than those obtained from wild-type mice. So neurospheres, these would be small little spheres of these multipotent cells, I believe. So a neurosphere is a culture system composed of free-floating clusters of neural stem cells. So yes, so those are the neurospheres, and they provide a method to investigate neural precursor cells in vitro. So if you have a neurosphere that is isolated from a negative mouse of Galectin-3, there will be weaker commitments or less differentiation into these oligodendrocyte or oligodendroglial cells as you would with these Galectin-3 positive cells. So additional findings by our group indicate that glial-derived Galectin-3, but not Galectin-1, promotes oligodendroglial differentiation. On the whole, Galectin-3 contributes to myelin integrity and function, which has critical implications in the recovery of inflammatory demyelinating disorders. So here would be a schematic. So this is describing something that is written later, but this is a schematic illustrating which shows the different effects of transferrin and Galectin-3 and iron on oligodendroglial differentiation. So the Galectin-3 participates in the conversion of M1 and M2 microglial phenotypes. Galectin-3 secreted by the microglia induces the oligodendroglial differentiation. Apotransferrin, which is the transferrin that does not have any iron in it, this induces oligodendroglial lineage specification and also favors oligodendroglial differentiation. So iron deficiency increases the number of oligodendroglial precursor cells and also diminishes oligodendroglial maturation. So even though there's more of the precursor cells, there is less of the mature form of oligodendroglial cells. So this is indicating that the intrinsic OPC oligodendroglial precursor cell problems and the absence of the factor secreted by astrocytes or microglial affect OLG differentiation. So blue and orange arrows illustrate previously published crosstalk between astrocytes, microglial, and the oligodendroglial precursor cells, which positively modulate OLG maturation. So here, this is the direction of specification, right? And so both Galectin-3 and transferrin can upregulate these neuronal stem cells to turn into these oligodendral progenitor cells, right? So that can happen. And then these astrocytes have all these different growth factors that can theoretically cause them to, are these also being able to, they might be able to turn into oligodendroglial cells in of themselves too, but I'm not sure. Regardless, transferrin and Galectin-3 have the ability to help with this differentiation from the oligodendro precursor cells into the oligodendroglial cells, which can do the remyelination. So that's one thing. Now, granted, transferrin, it doesn't need to be transferrin in and of itself. Oh, and I should say Galectin-3 also has the ability to turn these microglia from the resting M1 to the activated M2. So this would be one of the positively pro-inflammatory responses, right? So it can do that, and this activation will release interleukin-10, which interleukin-10 will also help with this OPC to mature into this oligodendrocyte. So that's how Galectin-3 is playing its role, and transferrin is also playing a role, and it appears to be necessary for this role. So it's not that it's just helping, it is necessary. And granted, you do not need transferrin. You could use apotransferrin or transferrin without any iron, but the thing is, the iron is usually necessary to get transferrin in of itself. So, what I should. So yeah, and here you can see iron deficiency. So iron deficiency will actually increase this OPC but decrease the OLG. And I should say that iron is actually necessary for transferrin translation. So the transferrin mRNA is always going on, or I should say there's a lot of transferrin mRNA normally, but the problem is this transferrin mRNA has a protein that is bound to it that doesn't allow it to be translated easily. So iron actually will come in and remove that protein, which then allows the transferrin to, I might have it backwards. Transferrin, you want, yeah, I believe that's it. And so you want more transferrin the more iron you have. So yeah, the iron will come and bind to that protein that removes it from the mRNA, which then allows more transferrin to be synthesized. So, you know, I'm just going to do another video on this because this is way too much to do all in one go. So, with that, I'm going to stop right here, and there will be a part 2 that comes out tomorrow. So thanks for listening, and hopefully I will see you tomorrow.